I am Glenn Johnson, interim chair of the Department of Environmental, Occupational, and Geospatial Health Sciences at the CUNY School of Public Health. And I am very happy to welcome Dr. Cristina Davis as our Grand Round speaker for this month. Dr. Davis is a professor of mechanical and aerospace engineering at the University of California, Davis, and the UC Davis Associate Vice Chancellor for Research. Um, Dr. Davis's research group focuses on creating miniature analytical sensor systems for mobile chemical detection platforms. She earned her Bachelor of Science at Duke University with a double major in mathematics and biology, and completed her PhD in biomedical engineering at the University of Virginia, focusing on novel biosensor research. She then worked on silicon chip-based biosensors during a postdoc fellowship at Johns Hopkins. Along with a group of co-founders, she worked in industry in Switzerland to establish a startup company developing memes-based screening platforms for the pharmaceutical industry, and their company was acquired by Molecular Devices Corporation. Dr. Davis ultimately went on to become a principal member of the technical staff and founding group leader of bioengineering at the Charles Stark Draper Laboratory in Cambridge, Massachusetts. She has served as a member of the Scientific Advisory Board for the United States Air Force, is a fellow of the American Institute for Medical and Biological Engineering, the American Association for Advancement of Science, and the National Academy of Inventors. She is a co-founder and scientific advisor to a UC Davis affiliated startup based on her research. She was chair of the International Association of Breath Research from 2021 to 23 and is currently the editor-in-chief of the Journal of Breath Research. So with that, I welcome you, Dr. Davis, and I'll turn it over to you.
Thank you so much. I am delighted to be here with you today, and I want to thank you for the opportunity to tell you about the research going on in my group. As Glenn mentioned, I have an administrative role on campus, but I'm still very research active. And so, what I'm going to talk about today is what my personal research team is doing. And specifically, we're making tools and new infrastructure and architecture that can help epidemiological studies that some of you might be interested in, or the topics could be of interest to you. And we're looking at ambient exposures to volatile organic compounds. And I'll kind of get into a little bit more of this today. So just as an introduction, we have lots of chemical sensors in biology. When we think about detecting trace amounts of chemicals, we have our noses, a dog's nose, we think of that as being very sensitive to smells. But there are other chemical sensors as well, like the tongue is a chemical sensor for taste. And plants can environmentally determine if there are chemicals in their own environment, and they can respond to it actually. And then other animals have very exquisite sensitive smell. And the one in the lower left-hand corner is actually an African giant pouched rat. Those are sniffers that are really lightweight animals, and they're being trained to detect explosives in conflict areas. So they're so lightweight they can scamper across the ground and they don't set anything off. And so they're able to alert and tell people where the landmines are buried so that they actually can remove them safely so that people don't get injured. It's another chemical sensor. And then, even unusually, we can think about chemical sensors like the Komodo dragon. They don't sniff the way we do. They stick their tongue out, they sample chemicals in the air, they withdraw it into their mouth, and then they have something called a Jacobson organ in the roof of their mouth, and they shove their tongue up against it, and that's how they sniff. Each one of these is a chemical sensor. So we're surrounded by them in biology. So you would infer from this that they probably are important ecologically for animal species. And so we can imagine that we may be able to use that information as humans as well. And so when we think about that, there's a challenge in front of us that are a couple of ways to frame it. One is, what are the chemicals that are of interest? So, on the far left-hand side, I have this fingerprint of chemicals. Like, what are the constellation of chemicals that are very important to know? They could be things that we already know, like things that are environmental toxic hazards. It could be things that are biologically produced and they're biomarkers, but there's chemicals to be detected. Then we have a hardware problem where we need to develop a sniffer, for lack of a better word. And my group has worked on this as well. And we can sniff these chemicals in complicated environments, and they generate really very complicated data. And that is kind of that second to the right panel where we think about the data comes out and it's very complicated. So you can't just easily, quote, look at it and see what it means. You need some kind of software system or ability to interpret that data. And then finally, you need a way to turn this into an actionable like event. So when you've sniffed something, your instrument recorded it, your data told you what the result was, and then you need to have some outcome that happens from that. So this is kind of the paradigm that my group has been operating on. So I have kind of two halves of my research team. Half of them are engineers, and the other half are chemists. And the engineering team works on the sensor development, they work on the software. We're using artificial intelligence and machine learning algorithms to be able to interpret the data, but everything around kind of the detection and interpretation, that's falling under an engineering side of our house. The other part of my team is actually chemistry, because you have to have a pretty good understanding of that to build the sensors appropriately. And we also do gold standard work using confirmatory mass spectrometry to determine what the chemicals of interest are. If it's not something that's given to us like the EPA, where they would say, like, here's hazardous things, don't get exposed to them. Unless it's from that, we sometimes need to determine what the chemicals are. And that's some things that our chemistry group does. I do need to give you guys a conflict of interest disclosure. As mentioned earlier, there's a startup company that spun out of my research team. I don't hold a position at it, but they have licensed, issued patents that came out of my research team, and those are listed here. One or two of them have been licensed to other companies as well, and those are also listed. And I have sometimes been a scientific advisor to them. I report on this annually to the leadership on my campus, and I have permission for the activity, but I'm required to tell people about it since I'm going to be presenting some of the things that have been licensed out to industry today. I also have a without salary appointment at Los Alamos National Laboratory and also the Veterans Administration of Northern California. So I have a really long-standing clinical collaborator. His name is Nick Kenyon. We have worked together for almost 20 years now. So I joined the UC Davis campus November 1st of 2005. And in January of 2006, we started talking. We went to have coffee. We were both assistant professors. We had an interest in the area of human diagnostics, and we started working together. And it's 20 years later, and we're still like going strong. And we've had joint students. We've had grants that we have co-written together. So before I even get started, I just want to talk about this was a foundational collaboration, and he has been a partner throughout all of the work that I'm presenting today. When we think about health relevant information, whether you're thinking about it from a public health perspective or Nick is thinking about it from a clinical diagnostics perspective, or I'm thinking about it from an engineering perspective of things we can detect and measure, all of this is easier if it's non-invasive. And so we look at health relevant information that are coming out of the human body in a non-invasive way. And these are smells, basically. So odors that come out of the human body, whether it's emitted through your mouth and you're exhaling it, or whether it's emitted through your skin and you're sniffing it. They each give us clinical insight as to the whole body metabolome changes that could be occurring, whether it's disease or sickness or exposure to something. And these biomarkers are either volatile, so they're odors we sniff, or they can be non-volatile compounds that are usually typically measured in blood, but they can pass the blood membrane barrier in the alveoli in the lung and or in the skin barrier for the dermis, and they can be emitted from the body, and then we can measure them. And so in the breath, those non-volatile compounds are in aerosolized droplets. They're not gas phase anymore, and they comprise a whole lot of different things. And I'll talk more about the types of biomarkers that we see in a little bit. So we aim to use non-invasive monitoring as a way to sample and detect things because it makes an easier burden for clinical studies as well as for the patient. So the idea of looking at exhaled breath is not new. In the 1700s, two French scientists, Lavoisier and Laplace, made the first step in understanding what was in exhaled breath because for a long time before that, people thought, I inhale air, I exhale air. They didn't really understand that we were inhaling oxygen and we were exhaling carbon dioxide. And what they did was a fundamental study where they had this patient over here on the right-hand side exhale through a mask. They had him blow through a tube that bubbled through a bicarbonate solution and then came out the other side in a water bath. And what happened was the carbon dioxide built up in solution and eventually hit the point where it precipitated the bicarbonate so you could physically see that it was present. Believe it or not, this was the first time that we realized, scientists realized that you were exhaling carbon dioxide. You were exhaling a metabolite. And we take this for granted now. This was the scientific advance at the time. We have much better instrumentation now. So when we think about these exhaled breath metabolites, there's kind of two fractions. There's the gas phase and the non-gas phase. So the gas phase is things I already mentioned, like carbon dioxide, but also things that we also now know about, like nitric oxide is a commonly known exhaled breath biomarker. There are all these small compounds called volatile organic compounds. They're tiny and they have a lot of interesting relevance. And those compounds are numbered in hundreds of them in our exhaled breath. The catalog of what's in breath is up to about 1,100 of volatile organic compounds right now that we can identify and put a name on. And some of those are at higher concentrations than others, and we're still trying to figure out what they mean, but we can measure them, and there are things that are so trace that although normally they would have an odor that you could perceive, even humans could perceive it, dogs definitely can, but it's below our threshold of perception, so we need advanced sensors to be able to do this. That's the first fraction, gas phase. The second fraction is the aerosol phase, and this is where we're looking at those aerosols that are created usually in the lungs. Some aerosols come out of our oral cavity, so there's some saliva that gets aerosolized. That's not really interesting. It does have enzymes in it, but we don't, unless you're interested in those enzymes or saliva metabolites, you can kind of ignore those. And we look at the lung aerosols. Those are tiny droplets that are created usually in the alveolar space, we believe, and then you exhale them. They can have infectious disease agents in them. And so this was a part of the pandemic where we all realized that infectious disease spread in aerosolized droplets is something of concern. But in addition to what could be in there are things like inflammatory biomarkers, large compounds that are normally found in blood. You can see drugs in breath, and all of those are in that aerosolized fraction, but you have to sample it correctly and you have to analyze it correctly. And then the last thing is what we call exhaled breath condensate. This would be if you hold a mirror up to your mouth and you blow on it and you go, that creates a condensate film that you can see on the mirror. Now, imagine doing that in a way where you can actually capture that condensate. It is a liquid fraction. It has some of the aerosols in it. It has some of the volatile gases in it, but it's in a liquid phase and it's in a film. And we have a sampler where we can actually collect that and then analyze that using benchtop or mobile analytical equipment. So just setting the stage for where we're going with all this. So we created a silicon chip that can actually take that sample of the exhaled breath condensate. It's a micro electromechanical system or a MEMS system, very similar to the types of chips that you have in your cell phone, and it has topography in it. So you can see all these little tiny features. This is a scale bar down here, about 500 microns for this fiduciary mark. And each one of these is a little physical ridge that is in a sampler. What it does is it changes the topography of the chip so that when you exhale onto it, you can actually capture those miniature aerosolized particles. You can actually condense some of that liquid coming out of our breath into a format that we can actually work with. So it looks like this. It condenses into these droplets. They range from small droplets to big droplets. And you'll notice that they roll around on here, and that's by design. We actually created this topography with a specific purpose in mind, which is to collect the condensate, accelerate that condensation process, get the droplets to roll towards the center. And we do this on a super hydrophobic surface so that we can collect all these at one time. We can enhance the amount that we collect, and then we can analyze it biochemically. This shows an example of what that chip looks like from the side angle. And this is a very large droplet that's big enough to see. And you can see we're manually placing it. This just shows you the device itself is super hydrophobic, which means that the water droplet kind of beads up, very much like in the morning when you see dew on a plant that's out in your yard, you'll see it bead up on the top of the surface of the leaves. And that's that same kind of topography that's on the leaf surface is on our chip surface. So we're condensing the breath, we're getting it to roll towards the center, and we're doing it quickly enough that we can collect all this precious sample into one spot so we can grab it later and then analyze it. That's kind of the goal of what we're doing. This shows another different sideways view of the time course of the droplet movement. This is interesting because we engineered the topography of that chip so that the drops would roll. And you just saw it roll. So we know it works, but how does it work? And the way that it does is that the topography has an increasing wetness angle that as it goes towards the center, it drives the droplet to roll. And it does that because the contact angle, I'm going to pause it for a second. The contact angle is that angle between the edge of the droplet and the surface. You can see that angle all the way on the left-hand side is pretty big. And over here on the far end, I can't stop it at the exact point. It's much less. So you can see that this angle right here is actually a more acute angle. And so by driving the droplet through this wetness gradient, we're able to drive it towards the center. We do this through engineering. So we know the math formulas of what makes it roll. We're able to kind of confirm the topography of the chip so that it actually rolls in a certain way. And the contact angle actually decreases as it rolls from right to left. So it kind of looks like a full tire and a flat tire by the time it gets to the end. And those droplets are those exhaled breath condensate droplets, and that's got biological content that we want to measure. So we took one of these chips and we put it into a device that we could breathe through. And this shows one of my PhD students, and he's breathing into the device. It's got a little battery that's handheld so that we can actually control like the power requirements of it, but it's portable. And you can inhale through your nose or your mouth, either one, and you exhale through this commercial device. It's a commercial mouthpiece that we buy. that's inert. It goes through this little twisty bend area, and this is an aerodynamic filter. So as little droplets go through here, they're so little and lightweight, they slip right through this pathway with no problem. But big droplets like saliva or spit, they get filtered out and they slam into the side and they collect here in a spit trap, basically. And so the rest of the little ones go through, they impact onto the collector, they roll like that droplet I just showed you, and then they collect on the surface of this chip inside this cavity of the device. And so we did experimentation with this with patients to see if they were able to take their own breath samples in their homes. Because this is great if you could do it in a laboratory, but it's more helpful if you can do it in a private environment like somebody's house so that they could actually take clinical samples in a more austere setting. So what we did is we did a pilot study with a bunch of teenagers. We figured that they would be the toughest patient population to work with. So we had a bunch of 15-year-olds. We gave them our devices, asked them not to break it if they could. And then we basically sent it home with them, and they took samples from themselves every 12 hours. And they stored the liquid that they after they had the liquid exhaled breath condensate, it was it turned into a big giant droplet on that surface. They poured it out into a vial, stuck it into their freezer, and then we came and picked it up at the end. So they took every 12 hours for 2 weeks. They took an exhaled breath condensate sample. And these patients were either healthy or they had asthma. And you can see over here the outcome. We were excited to see this. All of the asthmatics clustered together when we look at their metabolomic content, and all the healthy controls are green. There was one that had more divergence than the other patients, but which we don't know why this one had so much variability. But it's very clear how to separate these. So once we picked up the samples, we lyophilized them, we ran them on a liquid chromatography mass spectrometry QTOF instrument. It's a high resolution mass spec instrument. We looked at either targeted or untargeted metabolites. For the targeted metabolites, we can look at things like inflammatory markers because we know that asthma is an inflammatory disease. It's also exacerbated by environmental insult. And then we also had untargeted where we could actually just see what all is in there. And then we did a statistical analysis on it. Kind of jumping through to just the end of what we found, each one of these pathways is an inflammatory eicosanoid pathway that's associated with upregulation of inflammatory markers. Many of these are disease pathways that are known to be associated with asthma, and some of them are drug targets. So, for example, the P450 pathway, which is this one over here on the far right on the upper panel, the P450 is a really common drug action pathway that asthma patients are sometimes given inhalers for, rescue inhalers. Now, some patients take systemic steroids, and that just suppresses all of their inflammatory markers across the board. But some of them are given very drug-specific pathways. And right now, the state of the art is you try a drug for about 6 months. If it didn't work, then you come back to your doctor and they try a different drug. So it'd be really great if we can see up front which patient might actually use drugs from one pathway over another because they have inflammation that's manifesting in a specific pathway. We have not done this yet, but we aspire to try some personalized medicine approaches to this so that we can actually tell whether or not somebody would be predicted to respond to one drug over another. What's interesting in here is you'll notice that in a lot of our asthmatics, most of them were on some kind of systemic steroid, and their asthma was well controlled. So you'll see that the inflammatory compounds that are represented here are diminished in amplitude in the asthmatic cohort almost across the board, except for this one. We don't know why this one was actually quite upregulated compared to the controls. But the diminished asthmatic profile is probably because of the systemic corticosteroids that they were on at the time. So this is really interesting because, again, asthma is a disease that is environmentally triggered. It can be things like pollen or things that somebody's allergic to, but they can be things that can be controlled, like PM 2.5 particulate matter that we breathe from uh, traffic areas or from wildfire events. That's something that we can control. We can have air filters for it. We can have masking if that helps a patient. But understanding kind of what's happening to their biomarker profiles is one way to record a response and to think about epidemiological studies where you could have an intervention and you could try to alleviate some symptoms of inflammation by doing some kind of targeted intervention. But you need some kind of an assessment to be able to tell whether your idea worked or not. So that's one of the ways we could see our work being applied. So I talked about humans, but you can imagine this works in animals as well. So we did some work years ago with the U.S. Navy Marine Mammal Program, and this is a picture of some of the dolphins. It's a group of animals that are actively managed by the Navy. They live in San Diego. They swim around San Diego Bay every day. They go out and exercise, and we had a task to try to figure out how to tell if they were going to get sick or not, because it turns out that they get sick and they can't really communicate and tell us why they're getting sick, and they also hide it really well. So sometimes you don't notice they're getting sick until they get really sick, and we want to keep them super healthy. So we figured out how to take samples from these animals, and you might, like anybody who's ever seen, marine mammals, they have a blowhole in the top of their head. So they inhale and exhale through their blowhole, and they eat through their mouth. This is great because also their exhaled breath has a disconnection from oral contamination. So we were able to get fantastically beautiful samples from them, even more so than what we were able to do with humans. So we took samples from these animals. We did it while they were either, this one you can see he kind of came up and sat on a mat for us while I took his sample, but sometimes they swim up to a dockside or they can swim up to a boat. And because it's non-invasive, we basically can just measure what they're exhaling as they kind of come into an area that we can access what they call blow, which is the exhaled breath of these animals. And we measured some of these animals for up to a year or 15 months at a time so that we could understand how their breath biomarkers changed when they got sick. So this shows an example of a few different case-controlled studies where we actually measured biomarkers when they were healthy. The red part shows when they got sick. This showed, in this case, a biomarker that was downregulated. Over here, you can see one that was quite upregulated. In this case, the animal was pregnant, but we also had some examples I don't have time to show today of some of animals that had upper respiratory infections or they had other diseases or disorders. So the breath samples that we collect with those animals are actually condensed through a larger device. It's not quite like the one we had for humans. And it turns into ice crystals, and this is what it looks like. And we are able to analyze that biochemically. So all of this fed into where we were in late 2019, where we were looking at a study to see if we could diagnose upper respiratory infections like influenza at a point of care, like an emergency department. And we could, if to see if we could sample breath and turn that into a diagnostic for the flu. So we were in the middle of this study, and March of 2020 rolled around, and we were in the right place at the right time. We had data that kind of indicated we were able to see differences between airway viruses, influenza, and controls. We were doing this in the emergency department at the time in the UC Davis School of Medicine. And many of you know what happened in March of 2020. The pandemic came around, and it became very urgent that we release our time so that the doctors could start treating COVID patients as they came in. And that happened pretty quickly. By April, our clinic was starting to see a pretty high influx of patients with community-acquired COVID-19 infections. So we halted our influenza trial. But then we also thought to ourselves, we're in the right place at the right time. We have an IRB protocol to collect infectious flu breath samples. We're already in the emergency department taking breath right before the pandemic broke out. Could we amend our IRB and could we start pivoting and looking at COVID? As long as it didn't become a burden on our healthcare workers like Nick, who was literally on the front lines, like he and one of our other colleagues diagnosed one of the first community-acquired COVID infections in the state of California and in the country that happened in March of 2020. So, right place, right time, we pivoted so that we could start looking at COVID. And in fact, we sampled for several years. Through the pandemic, we did see a breath signature in both the gas phase volatile organic compound metabolites, and I'll show you in a little bit some of those inflammatory metabolites because COVID is an inflammatory disease. So, what was interesting about this is we were measuring and measuring as time went along, and what we saw was the difference between the Delta and the Omicron wave of COVID that came through California resulted in a big shift in the signal for the volatile organic compounds that we were looking at. So it looked like this under Delta, and it had a very clear profile. It looked like this under Omicron, and within Omicron was so infectious that within a 2 week period, all of the Delta cases were gone and they were replaced by Omicron cases. That was really interesting for us because within a 2 week washout period, we went from one regime to another regime. We sequenced all of the nasal swabs from the COVID patients so we know what variant they had. And since then, we have not seen the breath profile of COVID change. So it's more or less, it's changed a little bit and it's got little tweaks, but it hasn't been massive. This was a massive change, and it happened at the same time that COVID was changing from being a lower respiratory infection to more of an upper respiratory infection that was clinically observed. So we know that these volatile organic profiles are very helpful in diagnosing, and we were also asking the question, what happened to that EBC condensate fraction with the eicosanoids and inflammatory markers? So we went back in with these same patients. We took breath samples from control as well as COVID. At this point, we were taking patients on the inpatient ward. So these were hospitalized patients that were on they were actually on ventilators most of the time. We took samples from them, but so they were no longer ambulatory. The ones that we took the volatile organic compounds from in the ED in the emergency department, they were ambulatory. These were hospitalized. But it's very clear across the board, every single one of these biomarkers, 1 through 15, is a different inflammatory pathway, and they were systemically upregulated across the entire cohort that we saw, and with very high detection capability. Now, we knew they had COVID. At this point, we have really good COVID diagnostics. And we all have lateral flow assays. If you think you're sick, you can go to the pharmacy and buy one now. But at the time, they didn't exist. And so we were doing this without any concurrent confirmatory tests except for quantitative PCR. So we still have ongoing clinical development in the area of breath, but I'm going to switch to a different topic for the environmental insult people that are interested in how some of these tools can be applied to other clinical questions. So we now have a branching point. That breath work for COVID that I mentioned, that was actually an emergency funded program under NIH that we were able to do. We had a multi-year grant during the middle of the pandemic to do that work. We now have a group, a program that's a 5-year UO1 NIH award to look at skin-based disease diagnostics, and it's a jumping off from that COVID work. And we've engineered a platform to collect and measure skin volatile organic compounds, so really tiny little odor compounds that get emitted through our skin. Many of us have heard stories about how dogs can smell cancer. Um, this is our chance to look at that. We have 20 different diseases and disorders that we're looking at with this instrumentation, and I'll show you a picture of it a little bit later. And we're in the middle of that. We're about to enter year 5 in June, so we're kind of coming to the end of that, and we're starting to kind of combine and coalesce all of our data so far. But back to environmental chemicals. One of the things that we know is that volatile organic compounds in the environment are largely unregulated, but they might have health hazards. And so we created a device that actually has a chip that's, again, like the types of chips that you see in computers and cell phones. And these chips they have little vias, little pathways that go through them where you can suck in air. It has a little sorbent in here, and then it's got little heater pads on it. You can suck in chemicals from an air sample. They stay on the chip. This is the sampler. It can be worn on a hip. You can wear them on a backpack. And basically, it's an environmental sampler that you wear around, and it can tell you historically in an integrated fashion everything that you got exposed to over the last 6 hours, 3 hours, 12 hours. You can set the duration for any time you want. And these chips become a forensic archival record of all of the incidental effects of chemicals that you might have gotten exposed to at any given time. So this shows the sampler. That's the little chip. It's got a lithium ion battery in there. It's got a GPS. And this is what the data looks like when it comes out. Every little thing here, every little peak is a different chemical, and the intensity and abundance of the chemical is higher or lower. So we can quantitatively see what was there. So we use this in a few different ways. You can see this is just showing that the GPS worked as somebody walked around on campus, but we also showed it worked in a few settings. So this would be like sampling ambient volatiles while you're cooking. This is another one where somebody walked down a hallway in a building as the floors were being waxed, industrial like chemicals, solvents, and then also just latent environmental chemicals that were around in an environment. We did this work back in 2019 to try to see if we can quantify what people were getting exposed to. In California, we have wildfires pretty regularly. You guys might have seen in the news the one in LA that was especially large, but we have small wildfires all the time. So we also tried taking some of these and putting them onto UAVs or unmanned systems so that we could fly them around and we could test the air quality in different locations while an event is occurring. And so we did this in 2018 with the campfire. That was the one that was in Paradise, California, that was, you guys might remember, was really bad. And then we did some testing just to get our instrumentation ready with our local fire department. But this data is actually from the campfire that occurred in Northern California. This is really interesting for lots of communities. I think environmental toxicologists are interested in this kind of data. It's very hard to determine what gas chemicals people might have been exposed to EPA doesn't have regulatory thresholds for all of the chemicals. They have some. And there's not a lot of instrumentation that can actually tell you historically what you were exposed to at different time points. So we think that this kind of forensic evidence could actually feed into epi studies that could begin to test health effects of what all is going on. This is what our chip looks like. For engineers, this is really cool because we made these and tested them once, but then we also made big batches of them, and we showed that we could manufacture them in relatively high quantities, like in 100 or 200 at a time, so that we can actually ship them out and do epidemiological studies with them. So that just shows some of our work. So for us, fire is one of the largest environmental exposure studies that we might be interested in. We have a group in LA County that is with the LA County Fire Department that we partnered with, and they were interested in knowing what chemicals are our firefighters getting exposed to from an occupational health and safety perspective. When they go out and they are doing their job as a firefighter, what are they getting exposed to? Obviously, it's combustion products, and that's going to vary depending on the source of the combustion. But how much does it vary? What did each of their shifts cumulatively acquire in terms of their exposure limits? So we took some of our chips, you can see four of them here, we housed them into a new package and we mounted it onto the pump panel of a fire truck. So you can see it up here on the upper right-hand side. And basically this sticks with the fire truck, and the firefighters never go that far from the truck. So like it's a proxy for what they may be exposed to. It actually can collect on four individual samplers, so we can set it so that it'll do 6 hours on chip number one, and then that turns off, and then we do the next 6 hours on chip number two. So you can get a time sequenced average of what chemicals the firefighters are being exposed to at any given time over the course of 48 hours. So each one of these little boxes can go for as long as 48 hours. We usually had them set for 24 hours. And these are portable. They go with the fire truck, so we also have GPS location. This shows an example of us showing the GPS location work. This was some testing around campus. This shows an example of how we desorb it. Once the chip has accumulated its sample, we save it and then we send it back to the lab. And right now we're off-gassing it into a mass spec so we can actually measure all these tiny little chemicals. But we have a version of a real-time chemical sensor that's in progress. It just wasn't ready for the fires that we had in the last year or so. So in 2024, our LA County fire partners had some wildfires that they responded to. One of them is shown here. You can see their GPS locations. That it's actually not finally resolved GPS because to protect the privacy of the places and the homes where people would have maybe been going to respond to for the firefighters. This is in review at military medicine right now. And you can see each one of these profiles has lots of different chemicals in it. This is just data up here showing us that it worked properly. And we measure things you would expect with combustion, but it's a lot of different kinds of combustion. We see some chemicals that are called BTEX, that's benzene, toluene, other xylenes, other chemicals. BTEX are associated with combustion events. We also saw PAHs. We saw aromatic compounds, hydrocarbons, and we're working to quantify target compounds to understand what the exposure insult was for the firefighters at any given time. So we have some collaborators in our public school of health, and they're looking at the health assessments for the firefighters, and we're just one measure. They have other measures that they're looking at. So I'm not going to speak about their work. where they're trying to understand other measures that could tell us information to help the firefighters understand and control their own health information. But this is just a big list of what we saw in the air, and we know that they were there. So if they didn't have a mask on, they were breathing it basically. So I'm going to go back to something I mentioned earlier, which is we have a skin program funded by NIH right now to look at health-related emissions for volatile organic compounds. This is my clinical collaborator, Nick, in the right-hand side, and he has a wearable device on. This is our device over in the corner. It's got a lot of wires hanging out of it. It's not really ready for prime time use, but we have it in the clinic. We're generating data, and it has real-time measurement of volatile compounds that are emitted from skin, and we're looking at diseases and disorders. So I'm kind of looping back to stuff that I mentioned earlier in my talk. And you can see us also taking ancillary measurements as well, like things like heart rate monitoring and some other things. They're all built into this wearable device. So the device that's the real-time detector is called a differential mobility spectrometer. This is a chip. It's about the size of a quarter. This is what it looks like right now. It's also like small microfabricated, like you have in silicon semiconductor chips that are in cell phones or laptops. This one has chemicals that come in through one side. Going to skip through some stuff. The chemicals go through the heart of the sensor and they wiggle around in an electric field. And the way that they wiggle tells you the identity of the chemical. I apologize for making that really high level, but it's pretty complicated from a chemistry and physics perspective. But we can get information out of the chemical sample that tells us what the identity of that chemical is. The way we're getting the sample from the humans is we have a patch that actually sits onto the forearm as a slightly elevated, heated line of gas that comes into it. It gets the skin emissions and then routes it into this instrument for real-time detection. This is what some of our data looks like from 4 different patients. Each one of these little peaks is a different chemical. So we can see a little over 200 different features in some of these, and we're looking right now to understand what the difference is between them. In some initial data, we're starting to see differences from skin VOCs between our healthy and disease cohorts. This is really early data. Frankly, we need a lot more before we can make any conclusions that are all. We have used the same instruments to look at exhaled breath, though, and we're using them as environmental samplers as well. So I wanted to make sure I got through most of my talk. I'm happy to answer questions, but here's some thoughts about where we're going with it. We're making it wearable environmental samplers. We think they can be used for epi studies in the future. We started a little bit of that with the firefighters, and we're going to continue that work, maybe with some other occupations as well. We're at an early stage right now. We're also looking at exhaled breath as a measure of pulmonary inflammation. This could be from upper respiratory infections. We hypothesize that we could do this for environmental insult as well, like PM 2.5 exposure from wildfires. We know that there's a clinical uptick of patients who come in with upper respiratory ailments after we have large wildfire events in California. And so being able to track exposure to clinical data outcomes would be very helpful for us. It would also help us to think about regulatory limits on the kinds of environmental emissions that could trigger health effects in humans. And those limits are not always well established. And then finally, we've put these on unmanned systems and flown them around, but we also have some ideas on putting them on vehicles and driving them through locations and areas so that we can monitor VOCs across more large spatial differences. So I want to thank some of our funders. We've had NIH funding for the skin work and the breath work. We also have had some NSF funding on our sensors, as well as Department of Defense funding, and then some other folks that we've been able and fortunate to work with over the years. And then these are the folks that actually do all the work. So I have to, give a huge shout out. Mitch McCartney is the director of research from my group. When I'm off doing some administrative stuff, he's the one that keeps all the trains running, keeps everything going. And then I have a variety of students that are both grad students, undergrads, and then we have some postdocs and staff that are in the lab. So with that, I will stop so that I can answer some of your questions. But it has been really wonderful to tell you about our work today.
Wow, that's that's a lot of great science, and I the diversity of the applications is what I'm going to really carry away with me. From human, asthma, COVID, to wildlife science with dolphins, to wildfire for overall environmental health, to firemen, women, working, the occupational aspect, just so many applications. Um, Bill Pockles has some questions that he's listed in the Q&A. I'm not sure if you can read that, Dr. Davis. If not,
I
if
Oh,
not,
wait. Yeah, there it is. Have you heard of the nasal ranger? Um, and that one is a new one for me. So I haven't heard of that one. I'll have to go look at it. Do you want to unmute and say anything about it?
uh, Emmanuel, do you have control over that to allow people to speak?
If you guys would like to. Otherwise, you can ask more in the question. I didn't see the Q&A at first, but I can
Oh,
see it now.
okay. It's Emmanuel is indicating that the attendees may not be able to unmute. So,
Okay.
um, but then one question Bill asked, the kind of practical is would copies of the presentation be available?
So I sent a PDF of our PowerPoint to Emmanuel, and I'm happy for you guys to
Oh,
share that non-publicly, please. So don't post it on the internet, pretty please. And
okay,
and it's mostly published material, so I'm not
right.
worried about that. If you're going to circulate it, Emmanuel, I need to give you a copy that has some of the Department of Defense work taken out of it. So let me resend that to you later this afternoon. The PowerPoint is so big with the movies in it that it's not easy to circulate. So I'll send
Right.
I'll send a revised PDF to Emmanuel.
That's wonderful. Yeah, that's fair enough for sure. Bill also had a question. Did you distinguish between smokers and non-smokers? And
This is a great question. So in the state of California, and I'm going to stop sharing for a second. I'll share again if I need to. Um, in the state of California, we have a funding agency called the TRDRP, Tobacco Research and Disease Prevention Program. And it basically takes a box tax on cigarettes and puts it back into research for tobacco use. And we had funding from them to look at vaping, because so that's the poor cousin of cigarette smoking, where there's a perception that maybe vaping is not as bad. I'll tell you, it is just as bad. I'm sure many people in this audience would appreciate and understand. But what we looked at was, could you take exhaled breath from vapors, as well as potential recreational drug users like marijuana, which is legal in the state of California. And we did that. We published our work, I think, about a year ago, and two things came out of it. One is that we do see environmental insult that upregulates inflammatory pathways. The other thing, and this is stuff that's not published yet, but we're going to probably in the next 6 months, You can measure all kinds of things in the gas phase of what people exhale, and it has things like metal and things that are from the emission source in the vaping location. And so the content of what people are breathing in is much more than just the tobacco or the marijuana that they believe that they are ingesting. And some of those are potentially quite toxic, especially the metals.
Mhm.
That we have not published yet, but it's coming.
Excellent. And Professor Solomon has a question for the high resolution metabolomic in humans. Did you only use breath, or did you also use blood, saliva, and urine? And if so, did the biosamples vary?
So in the exhaled breath condensate, we've done LC-QTOF as well as other high resolution LC mass spec methods. And we're measuring mainly the eicosanoids in that fraction, but we do have some untargeted work that's ongoing. With the gas phase, we've done GC time of flight mass spec, and we have a new GC-GC time of flight that's coming online. Each method, we're measuring different kind of chemicals. And I think what was the other part of, I think I jumped around for part of the question was
Did the if so, did the biosamples vary?
yeah, we did one study. I mentioned drugs and breath in passing early on. We, a couple of years ago, we did a study with patients that had sickle cell disease. And sickle cell patients, much of the time they're healthy, but they can experience a pain crisis if their sickle cell, if they have sickle cells that actually lodge in different parts of their pulmonary system or in capillaries. And because of that, they usually can come into an infusion clinic and they are able to get painkillers as a part of their treatment plan. And so we were able to partner with one of my collaborators at the UC Davis Medical Center for patients who were coming in to get pain medication. It's IV and it's narcotics, basically. And so we were able to look at patients that had IV drug infusion for painkillers. We took breath samples and we took blood samples at different time points after they received their pain medication. They were opioids almost entirely. And we what we saw was within an hour to 2 hours, the opioids that were IV infused would show up in their exhaled breath, and then they would have a pharmacokinetic curve that looked very much like what you would see in blood. And so we took concurrent blood measurements at the same time because it's an outpatient clinic, so we were able to do a lot at the same time. And what was really cool is that as you see the drugs get metabolized, you see the pharmacokinetic curve go down in the blood, but you also see it go down the exact same way in the exhaled breath. It's just more dilute. So we were able to use breath condensate as a proxy for what we saw in the blood. And it worked with a handful of different opioids that different patients were on.
Ah, thank you for that. Next question is by Rachel Duff, which I find I'm curious about too. Are there any ideas towards developing an immediate readout of results as opposed to the GC analysis method? It'd just be incredible as a field monitor, something that.
Absolutely. And thank you for asking that. And I am going to go back to my slides for a second because this is an example of what our data looks like. We actually have a real-time detector. It's this guy right here. It's this thing. It's about the
uh,
size of a quarter, and we have it wrapped in an instrument. The instrument's not small yet, but it is technically portable. Um, it eventually will be small enough to be about the size of a brick. Not as heavy as a brick, but it'll be about that size. And eventually we want to shrink it to cell phone size, but that's a few years down the road. This is what our real-time data looks like. So this has a small GC column in it, and the detector is our real-time detector. And so this whole thing takes about 10 minutes to get a real-time measurement. Now, that's still not fast enough for some things. Like you want to know some things even faster than that. So we have a way to get, I'm trying to think, I don't think I don't have a real-time measurement in here. But we also have a real-time like seconds detector that is not deployed anywhere yet. But that's a great question because real-time measurement, I mean, that's where we want to be.
Okay. I want to skip the next question and come back to it because the question right after it, Mike, I think is a little bit technical and it also gets at something a lot of us are interested in. Have you been able to validate the instrument, the one you discussed in reference to firefighters in particular, with other validated VOC air sampling methods or direct reading instruments like a PID? Mike says they've done a lot of work measuring VOCs in nail salons here in New York City and have been researching validating low-cost sensor technology options to help workers and managers better understand exposures.
Yeah, absolutely. So we have some real-time GCPID instruments. We have some PID only instruments. So we do have those. We did benchmark against those. That part is not published yet, but it's in progress. And then the other thing that we've done is we used thermal desorption tubes. So I didn't know the mix of people you would have in your audience, but this is somebody that's really clearly does a lot of analytical chemistry. The chips that I showed you that are the sample chips, um, those are basically like microfabricated thermal desorption tubes. And so those were benchmarked side by side with actual thermal desorption tubes. And that data will be coming out probably about 6 months from now. We're working with our EPI partners, epidemiological study partners, to try to do that on the firefighter samples. That was where we did a lot of our benchmarking. And one of my collaborators used the wristbands. And I don't know if you guys are familiar with the silicone wristbands that are commercially available. Um, they work, they absorb chemicals, they're really non-specific, and it's hard to quantify things that come off of them. But in that paper that comes out about 6 months from now, we are going to benchmark our things, our methods against some of those gold standard methods.
Excellent. And to back up to a kind of straightforward question before that is, did you find any notable differences in exposures by race or neighborhood deprivation, which I think is being cited as just examples of different ways you could stratify the population you're looking at, and if you see differences.
Right. And this is exactly the question that we think our instrumentation can help, is that we suspect that it's possible that we would see differences. In our study with the firefighters, it was an occupational study, so we weren't looking at that. And even in the one where we had patients wear around the wearable version of our sampler, it was not in demographic cohorts. It was basically we gave it to teenagers again. They turned out to be a really good test case. We found teenagers are good at adopting new technologies. They were pretty easy on the technology. They didn't break very much. And then it was a good test because if you can deploy it with that group, you can probably send it out with other patient cohorts. But we didn't stratify it. But I think our technology could answer questions like what you're asking. We just don't have any data right now.
Here's a practical question from Mickey Siegel de Hernandez. He's interested in your occupational research, especially wildfire exposures. He says he works for a union with members in industries with chronic exposures to wildfire because of the work they do. Would it be possible to get in touch to discuss the potential for research similar to your research with firefighters?
Yeah, I'd be very happy to. So the department has my contact information. So if you want
Mm,
to send me an email, be really happy to talk because we're always looking for new areas where we might be able to do health measurements that can help populations. Very happy to
okay.
talk.
Okay. Yeah, that would be great. And I think, Bill Pockles too, who had questions earlier on about the nasal sensor and that. And he may also follow up to make sure you have
Yeah.
some of that information. And another question, any future application of breath analysis for THC?
Yeah, that's a great question, right? So we tried the opioids in breath, but that was really in a clinically defined population. So they were people who were in an outpatient clinic. They had infused opioids. They had IV infusions, so it was really controlled. One of the challenges with THC is that it is not controlled. And if you're looking at recreational use, you're in an environment where you don't know what the load was, basically. You don't know what the ingested amount was. The other thing that is also really tricky is THC is really sticky. As a for the chemists that are in the group that are out there, I think you guys will appreciate this. It just sticks in your instruments and it does not come back out for a while. And so it makes monitoring it very challenging. And the third aspect of this that's challenging is that there are not impairment limits that are legally enforceable right now. Part of that is a chicken and egg thing. If you have better instrumentation, you might actually be able to determine impairment limits, which don't exist right now, other than from blood impairment limits. But even those, I think, are mostly not written into state law. So we're not working on that. I have a colleague, friend of mine, that is working at NIST in Boulder, and she is looking at THC detection in breath. And I just met with her yesterday, and so there are people in the field that are doing it, but my group isn't doing that right now. So stay tuned, and I think you'll see some things from other folks in the upcoming years.
Mike, I already just followed up to thank you. And it says they've always been using TD tubes in the nail salons and haven't used the wristbands because of similar concerns that you've noted.
Yeah, we haven't used the wristbands, but it's widely known as they have some issues, maybe. And while you're on nail salons for a minute, I will mention something really interesting. Um, about 10 years ago, I did a study with one of my colleagues at Lawrence Livermore National Lab, and we were trying to understand if you could look at occupational health exposures through breath, because if you inhale things, you're going to exhale it eventually. And what does that look like? Can you measure it? And we looked at nail salon. We looked at participants at a nail salon. It was not the nail salon workers. And what was interesting is that the patient that went, the subject that went in and had exposure in the nail salon breathed out methacrylate compounds for about 36 hours afterwards. It was about a day and a half. And we took time resolved measurements of their breath samples, and you could see it decay pretty quickly, but it stuck around for quite a while.
Hmm.
And it was an interesting test of what might be possible with of exposure monitoring.
Yeah, Mike is one of our doctoral students who works closely with Professor Brian Pavlonis and done extensive work in nail salons and trying to, empower the nail salon owners to, just self-monitor, in
Yeah,
ways that, you don't have to put fear in them, like you're being mandated to do something. Just here's a device you can use that maybe trigger you to where getting elevated VOCs, maybe just open a window, turn on the ventilation, just to
yeah,
enable them to help themselves, So,
great ideas. Yeah, exactly. Ventilation goes a long way.
yeah. It's let's see if there's anything else. Kimberly Garrett has a question. Have you considered using these sensors to characterize PFAS exposure? I know that's Kimberly's specialty. I ask because of the application and firefighting context. There's increasing attention on firefighter exposure to PFAS through their turnout gear and tools.
Yeah, that question has come up, and in fact, it's on the list of compounds that we're looking through our data for now. And we bought the chemical standards, we ran the calibration curves. We don't have an answer yet on whether it is exaggerated. Uh, they probably have PFAS in some of their turnout gear. We know that is likely, but they also have emission sources when they have, especially fires at the urban wildland interface. They might be in a place where a car battery is on fire, or there are different sources of chemicals. So we are on the lookout for that. I can't say yet whether or not, I would say that's in progress, so stay tuned.
Excellent. Yeah, we have a little time. If anybody else has any other questions brewing there they want to share with Dr. Davis. It's,
I have to say, this is a great crowd. You guys are asking all the great questions. It's you never know what your audience is going to be exactly like. It could have ranged from clinicians for grand rounds all the way to chemists, and so it's a delight to get the questions from you guys. It also helps me know that I explained it well enough that you were able to ask good technical questions.
yeah. Thought the presentation was very clear, with such nice graphics and the diversity of applications. I think it's easy to keep your attention.
Well,
I,
thank you.
crossing everything we do, environmental health, occupational health, and this department is within a school of public health, and public health by nature is very cross-disciplinary. So that's why
Yeah.
you're seeing a mix of people in the audience. And it's one of the nice things about public health is that you get these cross collaborations and with people like yourself. And yeah, so
Yeah. Well, and the reason that I got into some of this work is UC Davis has a program project grant from the NIH. We have a P30 that is from NIEHS, and it's on one of my acknowledgement slides. And we're grateful for the center support. And it was through collaborations with people in our School of Public Health that we were able to start seeing how some of our work could contribute to this field. So,
yeah,
I think it just speaks to the interdisciplinary nature of it.
yeah. I can see going forward if you're partnered with epidemiologists to really design studies for exposed versus unexposed, that you could really carry this a lot further. And
Yeah. What
so
else?
one other question: have you compared to portable FTIR devices such as the Gasmet?
We have not. There's also some portable mass specs that are now on the market, and we have not benchmarked against those either. I'm guessing that we're probably at the same development stages, but I can't say for sure. So we haven't tried that.
Right. And
Someone
so
mentioned nutritional biochemistry.
yeah,
That's a really interesting one. We talked to somebody. UC Davis is a comprehensive campus. We have a lot of things going on, and we have a USDA site right off campus that has a nutrition group, and we have a department of nutrition as well. We've talked with them, but we haven't done any studies yet. But we were interested in seeing we would be interested in seeing, like, can you detect nutritional biochemistry changes that are affected in the exhaled breath, that you could non-invasively monitor. In the short term it would just be like a study, but in the long term you could imagine it would be a way to do preventative health, or personalized medicine approaches to nutrition. I think these are great ideas. It's just there's so many great ideas. We want to tackle them all.
yeah, yeah. So just another comment by Rachel Duff and Bill Pockles again. Have you compared your techniques to fast Fourier transform infrared detector? even though they're bulky.
Well, we haven't tried that. But again, like there's other instrumentation out there. The one thing that has not proved well in the field that I've seen at least are the eNOS sensors. And so we've avoided those. We like to have our devices help put names on chemicals. Would be similar to some of the portable mass spec instruments, but we haven't benchmarked against them. Yeah, but I think it's a good idea.
And then just another comment by Professor Solomon, just she's interested in the exposome via internal non-targeted metabolomics, so we'll be in touch. So,
Great. I look
yeah,
forward to hearing from some of
yeah,
you.
yeah. And as I'm sure you will, so share contact information, and you probably it looks like you'll be getting some follow-up emails, and hopefully
That's
it'll lead to some fruitful collaborations.
Yeah, I look forward to talking to some of you guys. I'm sorry I couldn't be there in person, but I feel like this has been a great format. Thanks for sharing your questions. I'll give Emmanuel another copy of my slides that have some redacted stuff taken out, and then they can be circulated. Please don't post them to the internet, but you can circulate them in your department.
Yeah,
And then really happy to set up Zoom appointments and talk with people one on one and see if maybe we can write some grants together.
yeah, it's that would be wonderful. And just one comment. You mentioned occupational exposures and doing some breathwork testing following those exposures. Is there a way, or could there be a way to backtrack for measuring PWAs for exposure?
I think so. We haven't, that's actually one that we haven't thought about too much. But I believe so. I mean, if you're interested in talking, I'd be happy to brainstorm.
So it's a little odd without having a room full of people, or or a screen full to just raise hands and that. But I think we're naturally coming to an end, and seeing thanks coming in, and I think a lot of people share what I've experienced too in the last hour. This has been very informative and very, very interesting. So
Thank you.
yeah, and we thank you too, Professor Davis, for giving your time. And I think it's
Absolutely.
fair that we could start signing off now.
Okay, thank you so much, and
Okay.
hope to hear from you all soon. Thanks again. Bye bye.
All right. Bye bye now.